This semester, in our biological inquiry class, our group learned a lot about nature through study of our study site. Let us take you through this wonderful world of biology.
Our site is located at the south end of SUNY New Paltz campus, in between South Parking Lot 35 and the Intramurals East Field. More specifically, the coordinates are 41 degrees, 73 minutes, 61.01 seconds north, 74 degrees, 8 minutes, and 80.81 seconds east. At least part of our site is used by and managed by humans, as it is an athletics field. The other wooded portion is left relatively untouched. Other defining characteristics of the site is that it is located near multiple dorm buildings and a parking lot. Natural characteristics of our site that potentially influence what type of organisms may live there is that it is a large, mounded, rocky area in the center of our site that is shaded, at least partially, by the trees from the wooded area. Some things that we liked about our site is that because of our site's unique location, which included a maintained area and a wooded area, was that we were able to have access to a wide variety of animals and organisms. What we disliked is that we definitely ran into some tough situations, both figuratively and literally. There were thorns everywhere in our site, and I'm sure we all have the battle scars to prove it.
Our region of New York is considered a temperate deciduous forest, and our site is host to a variety of flora, from the monocots on the lawn to the dicots growing in the bramble. The organism I chose to cover is the lesser burdock, which scientific name is Arctium minus. They're sometimes mistaken for rhubarb. It was introduced to North America from Europe sometime in the 1600s. The leaves are woolly and edible, used in a variety of foods and traditional medicine. They're dicots, so they have two leaves in the seeds, and they have hollow leaf stems. Every other year it flowers, typically between July and October. Pretty distinctive flowers, which are bulbous, spiky, and purple. And they have these hooked burrs on the flowers, which are used for seed dispersal, so they kind of catch on wandering animals and such. And those burrs are actually the inspiration for Velcro.
The plant I decided to study was Hesperus matrinalis. If you live in the American Northwest, you've probably seen many of these little leaves on the ground coming out from one point in the center. As it turns out, these leaves actually belong to a plant called Hesperus matrinalis, or Dame's Rocket, named such because a flower with a long stem will eventually shoot up from the center. Native to Eurasia, it was brought to North America in the 1600s and quickly dug its roots here, literally and metaphorically.
So, let's talk about bacteria. As we all know, microorganisms are living organisms that can only be seen under a microscope. Being that our site is an area with lots of soil, microorganisms thrive in the environment. Our team collected a sample of soil in February and diluted the soil bacteria. Then we each isolated a different colony from the agar plate. Then each of us streaked a plate and incubated them for a week. After incubation, we each picked a colony to gram stain and view under the microscope. My bacterial colony that I isolated from our group's streak plate was identified as Cytobacillus firmus, which is a species of bacteria within the genus Bacillus. This identification came from BLAST, which is the database that we use to compare the nucleotides of our DNA sequences. Something interesting that I learned after getting this identification is that the bacteria could be gram-positive or gram-variable, meaning it could be both gram-positive and negative. So it actually made sense that I was seeing both pink and purple rods under the microscope.
The organism that I chose to share today was the bacteria that I isolated from the soil sample my team and I collected from our site in February. When viewing my gram stain slide on a compound light microscope, I was able to see that my bacteria was gram-positive rods, and I also noted that it had an interesting budding pattern of the cells. When I plugged my DNA segment into NIH BLAST software, the outputted results indicated that my bacteria was likely one of the genus Bacillus, and was 98% certain that my bacteria was Bacillus thuringiensis. This is BT. BT is a common natural pesticide that is found in the soil. It makes proteins that are toxic to some insects when eaten, but not others, but is completely safe to humans because as mammals, we cannot activate it. And through my research of BT, I found that it has a tendency to form spores, and this was likely the activity I was observing under the microscope.
Thank you for coming with us on this wonderful journey through our study site. We hope you enjoyed it, and we'll be looking forward to what the biology department at SUNY New Paltz has to offer us next.